Yes. So good evening, everybody. So Taylor knows that I like to offer a moving target when I speak, so he's helpfully put a nice little white patch of tape on the floor. That is my limit. If anybody sees me moving out of there, you're welcome to wave and bring me back in. So my name is Ben Surridge. I work at Lancaster University, and I'm going to tell you something this evening about the Big Windermere survey. We've heard that mentioned a couple of times, so to end this evening's presentations before the panel, I'll just describe something to you about the Big Windermere survey. So, firstly, I should say, whilst I'm going to talk about the survey this evening, it's a project that's really relied on many partners, particularly the Freshwater Biological Association, who've been involved fundamentally in designing and delivering it, but also a range of other organisations, so UKCH that we've heard from already today. So what is the Big Windermere survey? So fundamentally it's a community or a citizen science project. So it's a project designed to investigate the water quality in Windermere, so within the lake itself, but also importantly in the streams and the lakes that drain into Windermere as part of its wider catchment. So when we designed the project we had two main aims. The firstly was really to see if we could engage the community in undertaking what you'll see are really spatially intensive snapshots of water quality. And really that was designed to try and complement exactly the type of monitoring that goes on in Windermere already, some of which you've heard about from Steve and Ellie at UKCH today. And then the second aim was, could we use that data and evidence from the Big Windermere survey to help inform conversations about changes, actions in the catchment that are designed to improve water quality. So over the last 18 months or 2 years that it's been running, it's probably grown to what I think is now perhaps the largest example in the UK of a citizen science project focused on water quality. We designed it in the early part of 2022 at Lancaster University with the FBA, and the first survey, some of you may have been involved, all those years ago, in June of 2022, was funded by Lancaster University. Many of you I know have been involved in subsequent surveys, and they've been funded by a range of organisations that you can see up on the slides here. So I mentioned this idea of a spatially intensive survey. What did that mean? Well, initially it meant that we identified over 100 sites from the very upper reach of the catchment, so the Rothay upstream of Grasmere, down to the Leven downstream of Windermere. So 100 or so sites where our volunteers could safely access streams, rivers, lakeshore areas and collect samples. It's really not a minor undertaking organising and delivering the logistics around these surveys. So you can see Emma and Trini from the FBA, who some of you will know no doubt, making up sample packs for our volunteers. So making up over 100 sample packs on each survey date that go out to our volunteers. There's also a slightly strange video some of you may see with me telling you how to take water out of a river or a lake. So the purpose here of the video and the packs is that we're all using exactly the same type of research approaches that I would use in water quality work at the university. Our volunteers all collect their samples within about a 2-hour window on the date of the Big Windermere survey, and they return them. I can see Steve cowering away in the corner there to one of our science hubs. So we have 4 locations around Windermere where these samples are returned, and Steve, to prove that he still does get his hands dirty and do some work, is here collecting and analysing data to begin with. Okay? Not just convincing you that zooplankton are your next best friend, but taking some water quality analysis. And then I will collect all these samples and return them to research laboratories at Lancaster. We'll undertake a whole range of water quality analyses in the research laboratories, and I'll tell you something about bacterial data today. And those bacterial analyses are done at external accredited laboratories. So we've actually run, I think Louise mentioned, 7 Big Windermere surveys to date. We started in June 22. The last one many of you were involved in, I know, was in February this year. So this overall then has been an effort of over 320 volunteers. They've given over 1,400 hours of volunteer time. And we've removed about 850 litres of water from the catchment and used this to complete now over 3.5 sample analyses. So I'll say something about the results of those 3,500 analyses today. And I'm going to cover 2 areas. I'm going to say something about nutrients, and then I'll finish by telling you something about bacteria. So I am a nutrient biogeochemist. I spend most of my working life on issues around phosphorus and nitrogen. I probably think it's fair to say that phosphorus has had something of a bad press recently. And like Steve and zooplankton, I'm going to try and convince you that phosphorus is actually a good thing. And we need to think positively about phosphorus. Why is that? Because we're all fundamentally reliant on it. If you want to survive as a human being, you will require phosphorus. If you want to build good bone structure, your bones are partly made of phosphorus. If you want good enamel around your teeth, I'm sure we all have a smile that looks exactly like this photograph. That enamel is composed of phosphorus. If you want to build the genetic coding system in your cells, DNA, RNA, you will need access to phosphorus. It is absolutely essential to all organisms. And those organisms include organisms that will live in rivers and lakes and streams. So Ellie and Steve mentioned cyanobacteria, otherwise incorrectly known as blue-green algae, within lakes and rivers. So I've shown you an image here of 3 different types of cyanobacteria. They will also require phosphorus. They use it to build their cell walls. They use it to build the genetic code within their cells. So phosphorus is really important. We will not survive without it. But as with everything in life, too much of a good thing causes problems. As you increase the amount of phosphorus available in a river or a lake, you can increase the rate at which these organisms grow. And you can also ultimately perhaps change the organisms that dominate in a community. And at some point that can lead to really undesirable consequences. And many of us will probably have blue-green algae or cyanobacterial blooms in our minds when we think about undesirable consequences. And you might be thinking about those on Windermere. And I'm just showing you in this slide that these type of blooms occur globally in fresh waters. On the left-hand side, I'm showing you a satellite image of Lake Erie. Okay, Lake Erie has the border between Canada and the US running through the middle of it. And the green colour you can see here is a cyanobacterial bloom on Lake Erie. This occurs really regularly and frequently and has some really significant impacts because the lake is the supply source for drinking water for the local community. And when those blooms occur, that disrupts public water supply. So the cartoon on the right, toxic algal terror, it came from the lake, is around in 2014 the city of Toledo, 400,000 people who were on bottled water for a period of weeks because they couldn't use the lake to supply drinking water. Just also wanted to say that cyanobacterial blooms are not solely an issue in freshwaters. This is the Baltic Sea. Here's the southeast coast of Sweden. This really rather beautiful green swirl image is a cyanobacterial bloom, this time in the marine environment. So phosphorus, as we've heard about today, is often seen as a major trigger for these type of cyanobacterial blooms. I should say to you that it really isn't the only factor we should be thinking about, and other speakers have talked about that. What's going on with water temperature and how often and intensely these blooms are triggered. What about other nutrients like nitrogen? But the target often falls on phosphorus. So I wanted to tell you about how good or bad the phosphorus concentrations are in Windermere. How high or how low are they? Okay? And I'm showing you here on the top panels for all of our samples from the North Basin, on the bottom panels all of our samples from the South Basin, and you've got 4 seasons, summer 22 through to spring 23. And we can use standards in UK legislation that comes from something called the Water Framework Directive to look at how good or bad the phosphorus concentrations are. So those standards allow us to class sites from high status in blue, good status in green, moderate in yellow, poor in orange, and red is bad status. There won't be an exam question on the colour-class relationship, you'll be pleased to know. Okay? The default target, where all of our waters should be, is at good or high status. So we should be in green or in blue. Okay? And before I tell you the outcomes, I must make it clear to you that the type of official classification for phosphorus that's done by organisations like the EA won't use these seasonal snapshots that I'm telling you about. They'll use, as I think Louise said, long-term average concentration. So there's a difference. There's a health warning on these classifications. But you can see, I hope, that throughout Windermere, in whichever season you look at, we are a long way away from having all of our sites in blue or green columns, in high or good status. The best performance in these data was actually in summer 22. About 60% of Windermere shoreline sites met that default target. In winter, that dropped to only about 10%. Okay? And indeed, you can see in the summer and the autumn a tail of sites in orange and red at poor or even at bad status, so really high phosphorus concentrations at those times. One of the really nice things about, I'll put my thumb up there, Taylor, I'll try to speed up a little bit, about the Big Windermere survey is we can compare Windermere to other lakes and standing waters in the catchment. So this is total phosphorus concentration from high to low across a whole range of lakes from Blelham, Esthwaite, Grasmere, Rydal and the two basins of Windermere. The orange diamond is the average concentration and the black dotted line is the average for the south basin of Windermere. You can see on average there's not much difference between these sites. Windermere is higher certainly than Grasmere and Rydal, about the same as Blelham, a little lower than Esthwaite. But individual samples that you can see as the black dots can be much higher in Windermere. Obviously, we have more sample numbers from these sites, but it is true that we see some really quite high phosphorus concentrations at Windermere, even if the average is similar. So in a few minutes, I'll also tell you something about bacteria. So we look at intestinal enterococci and E. coli, two groups of bacteria in all of our Big Windermere survey samples. These are, for us, fecal indicator organisms, a lovely subject I hope you've all eaten. For us, fecal indicator organisms are bacteria that if we see them at high levels in a sample, they suggest the presence of fecal material in that water. That's coming from our digestive system, but also the digestive system of animals, such as livestock and birds. And the concern there is whether that fecal material also indicates the presence of pathogens, human disease-causing organisms that cause sickness, for example, and diarrhoea potentially. So we can use bathing water standards to look at our bacterial levels between three different classes here. Excellent in blue, good in green, and less than good bathing water quality in yellow. Again, for the north and the south basin of Windermere, for 4 seasons from summer 22 to spring 23. Here we can see generally low concentrations of bacteria. Many of the sites would be consistent with excellent or good bathing water quality. But again, particularly in autumn and in summer, a tail of sites in yellow where we see much higher bacterial concentrations, sufficient to drop those sites below good bathing water quality standards. And again, we can look across the different standing waters or lakes in the catchment for E. coli on the top, intestinal enterococci on the bottom. The orange diamonds, again, are the average concentrations, and the blue dotted line is the average for the south basin of Windermere. Average bacterial concentrations are quite similar across these standing waters, but again, the individual dots, individual samples for Windermere can reach really quite high bacterial concentrations into these grey shaded rectangles, which are high enough to drop those sites below the standards for good bathing water quality. The same caveat and health warning with these data as I had for phosphorus. They are not treated in the same way as official bathing water classifications at the 4 designated bathing waters on Windermere are. Just to say finally around the bacterial data, one of the real strengths of the Big Windermere survey is this spatial intensity of sampling. So we can look across 100, 110 samples collected across the catchment at any one time and identify hotspots of poorer water quality. The CH and EA monitoring that you may hear about is fantastic for its timescale, its length of record. One of the strengths of the survey is the number of sites we can get to through our community science network. I'm just showing you one illustration of these hotspots. These are actually data some of you will recognise. This is Millbeck, not far away from where we're sitting at the moment, just before it flows into Windermere by the Windermere Jetty Museum. These are E. coli concentrations in Millbeck from summer 23 through to February 24. And you can see concentrations 6,000, 7,000, very, very high E. coli concentrations. To give you some context, the threshold for good bathing water quality for an inland water for E. coli is 1,000 colony forming units per 100 mils. So we are 6 or 7 times above that level. So there are definite hotspots of poorer water quality revealed through the Big Windermere survey. I could take you to other parts of the catchment, but Taylor will nick me off the stage before I can have a chance to do that. So, some take-home messages from me. And I was thinking about what are the two questions that I'm most often asked about Windermere. The first one is that people say to me they've heard about Windermere being ecologically dead. Is that the case? Is that true? So just some thoughts from me on that. Firstly, it's definitely true that if you go back to the 1950s, the 1960s in parts of the UK, there were freshwaters that were probably more accurately described as biologically dead. They were like many of the big industrial rivers in Europe that you might be thinking of. I wouldn't say that what we see in Windermere at the moment is consistent with that, but you've heard this evening about the pressures that Windermere, like many freshwaters, face, and that those pressures make that lake vulnerable to change, and we've heard about that this evening. Part of the vulnerability is definitely to do with climate, and you might think, well, that needs national or international initiatives, and I would agree with you. But some of those pressures are much more local, and they should be in our scope as a community interested in Windermere to grasp and to do something about. And Taylor asked me, what do you mean by community? And my answer to that is not just individuals. It's also about organisations that have a stake in that catchment, engaging and addressing these problems. The second question I spend a lot of time talking to swimmers and indeed to journalists about is the question of wastewater, bacteria, human health. So for me, I think it's important we're completely open and honest. Untreated wastewater is discharged into rivers, streams and lakes, including Windermere, at certain times and in certain locations. That's the nature of the wastewater system we have in this country. That wastewater can contain pathogens, and those pathogens do present, under certain circumstances, to certain individuals, risks related to human health. We have also heard, rightly, a lot about the importance of the infrastructure that water companies own and operate in relation to this issue. And I wouldn't want to negate that issue. That's not what I'm here talking to you about. What I would do, though, is to say we need to do more. And I can show you in the Big Windermere survey data evidence of wastewater and bacteria that have nothing to do with water company infrastructure, but they are to do with issues such as misconnections, private wastewater treatment works, septic tank infrastructure. So we need to actually combine a focus on infrastructure operated by the water industry with questions about what else we can do to manage wastewater within the catchment and reduce these things like high bacterial or nutrient concentration.